Good afternoon, everybody. Thank you for joining Ruben and I and Hamed for this journal club. Hamed Hosseini is right now a stagiaire in the group of Ed Gudmond at LYRIC, and he has chosen two papers which are quite small, but the topic is quite charged, and it's on cardiac structure that we all love and enjoy. It's the sinoatrial node, but whose anatomy and, let's say, functional anatomy, it's still quite a mystery, and it's quite remarkable how much we still debate on the nuances and the fine prints of how the sinoatrial node works. So, Hamed, thank you for proposing these papers, and I will just let you get on with your presentations. You will present two small papers, if I'm not mistaken.
Yes. Thank you, first of all, for the introduction. Let me tell you that in my stage, actually, one part of our job was about the SA node, and especially the conduction pathways that the SA node has, that recently there are lots of talks about it. And of course, this group, the group of Ohio, has done lots of jobs regarding that. And these two papers actually were the two last ones that I found. The second one is a research article, it's a research letter, and it's not a big article, but I have used the findings in that paper, and I've made the page about that too. Well, of course, the way that the SA node works and these connections are really, as you told, it is still there are lots of questions about that. So at first, I'm going to talk about the structure of the SA node according to this paper and the way of the function. Well, mostly this paper is focused on the way of the working of the conduction pathways and the way that it will matter in the arrhythmias and the way of the treatment. Well, after saying something about the structure and the way of the function, I will talk a little bit about the automaticity and of the SA node and the arrhythmia, the treatments that we can use. And please, if you have any question, you can ask it during my presentation. And of course, after that, we can have some of them too. Well, from the first days that the SA node was the question, there's a paper by Kielsen-Flach that they have found that the part of the atrium that is the connection between the SVC and the right atrium are the main part that makes the propagation for the heart. Well, in their paper, actually, they have two questions. The first one is that how the muscles of these part can make the propagation, and beside that, how can these muscles connect to the other parts? And this question is still not all of that all of it is not answered yet. So it is actually an intramuscularly 3D part, SA node, and it is located in the depths of the from 1 to 3 millimeter from epicardium. It is somehow discrete, and you can see the difference. And this is one of the pictures from the last paper. And as you can see, the width of the SA node is different in different parts of the atria. The parts that are more near to the crystal terminalis are a bit bigger, and the other side is smaller. But normally in the human heart, it's almost 11 to 30 millimeter in length, and it can be about a quarter or a third of the right atrium. And the width is 2 to 6 millimeters, and the thickness is almost 2.5 millimeter. And well, to find out the whereabouts of the SA node, there are lots of different ways to see that. First of all, as you can see, SA node is, beside the SA node, there are lots of fat that can be seen. This is another paper, an old paper, as you can see. This is a fat that can be seen here. But SA node is somehow tilted in a way that the superior part of the SA node is more toward the epicardium, and the inferior part of it is more toward the endocardium. Another way to understand the whereabouts of the SA node is the artery that runs through it. It is originated from the RCA. And in this way, you can understand to see the SA node easily. Well, one of the ways of seeing the SA node and differentiate it from the other parts of the RA is using the 3D histology-based models. Well, for the shape of the SA node, this histology easily can be done because it's so different from the other parts. But about the connections of the pathway of the connections between the SA node and the right atrium, you need more higher resolution and a smaller step size. And actually, this paper, they say that this really matters to understand how the conduction pathways are there. And they say that there are lots of papers that, because of the lower resolution, cannot see this. And they even say that this can make error in the results. For example, the one that you were talking about, Guido, from the I think Dr. Efimov. Yeah, there are lots of papers that say that SA node can be two parts. I was talking to Vladimir last week about that too. And these guys, the guys from Ohio, actually say that this is because of the errors that are in the way of the methods they are using. And well, the adult SA node is mostly, almost 50% of it is the fibrotic dense tissue. So because it is the fibrotic, you can use DE, delayed enhanced MRI, to understand, to see where is it to. And as you can see here, it is easy to see beside the fat that is beside the SA node, there is an artery that runs through it. And of course, there are lots of fibrosis inside. Well, the main actually thing that these fibrosis and fat are there, the main reason of their these fibrosis and fat is that they can protect the SA node from the hyperpolarization effect of the right atrium. And in the other slides, I'm going to talk about the other effect of the conduction pathway that this helps of the protection of the SA node too. In around 1990s, actually, the multi-electrode studies suggested that pacemaker complex can be too many, and they suggested that the existence of exit site can be there too. Well, with the advancement of the near-infrared optical mappings now, we can see that, especially with the different dyes that are now being used, we can see that there are some conduction pathways. They name it according to the places of that conduction pathways. For example, lateral superior, or the SVC, and the middle part. Most of this connection actually goes to the crista terminalis, but there are some papers saying that there are one maybe connection from the SA node to the septal part too. And well, the number of these conduction pathways are between 2 to 5, and except these conduction pathway, SA node is completely insulated from the other parts of the atrium. Well, as you can see in this picture, the conduction pathways are several discrete myofibrous tracts, and somehow, according to this paper and these guys, these conduction pathway works as an intermediate between the SA node and the atria. As you can see in this picture, even in the size of the myofibers, the SI node is smaller, and then in the conduction pathway, you can see bigger myofibers, and in the atrium and the crista terminalis, it is even bigger than that. And one of the main other reason of this is because of the problem of the source sink problem, and the way that one SA node place just a point maybe in the right atrium can make propagation to the other part of the atrium. And the best way to nowadays to understand the function of the SA node is near-infrared optical mapping, because you can see the intramural activation of the atria or anywhere you want in that. As you can see, the way it works is that the propagation starts in one of the compartments of the SA node, and then it exited through one or even two exits to the right atrium or to crista terminalis. And again, for example, as you can see here, exit through two parts of these conduction pathways can make this problem of maybe thinking of two SA nodes. And actually, one of the main things that this guy tells is that the location of the pacemaker site and the choosing of the conduction pathway are not correlated with each other. For example, in this paper, as you can see, the first place of the starting of the propagation is the lower part of the SA node, but the exit is through the higher part. Or in this picture, as you can see, it started in the middle, but it goes through the lower part of the conduction pathways. And this picture is from the last paper that I have sent, that I was trying to tell. The other way of working the function of the SA node that these conduction pathways help is the way of the myofiber orientation. They have done, I think, a really beautiful job. As you can see in here, the way of the myofiber orientation in the SA node part is somehow more parallel to the endocardium or epicardium. And then when it goes to the exit side, to the crista terminalis, it is somehow perpendicular to that. It means that the first propagation starts in here, and it goes to the crista terminalis, and suddenly from the crista terminalis, it moves to the endocardium or the epicardium. And the choosing of the endo or epi is not still understood, and why. Maybe it's just because of the size or the size of the CT in there, and the distance between the crista terminalis and the endo or epi in there. For example, in this picture, as you can see here, 10 millisecond, you can see that 10 millisecond earlier in the sub-epi than the endo part in here. So the only way that now we can think is that the myofiber orientation is in the way that it would passes easier. And I have found another paper. It was about the way of the passing of the propagation. It's an old paper, actually, by Rohr in 1997 about the improvement of the impulse conduction whenever you reduce cellular uncoupling. In this way, another way of helping the function of the SA node is the gap junction protein connexin 43 in the SA node. Actually, it is, you cannot find the connexin 43 that much. And also in the atria, there are lots of connexin 43s. And as these guys tell, the SACPs, the conduction pathways, again, are the something intermediate between these. And I noticed this old paper because, as this old paper says, when the place that starts the propagation, if the conduction in that place is small, it can make the conduction moves to the higher conduction part. So in this way, again, connexin 43 really matters, beside the myofiber orientation that I was talking in the last ones. Well, now I'm going to talk a little bit about the automaticity of the SA node, and I'm going to go through the arrhythmias in
Ahmed,
the.
sorry, let me interrupt you before you move to the function. If I understood correctly, there are no escapes to the epicardium preferentially, or it's because I saw only sections. So there are 3 to the crista terminalis, so it's intramural, and 2 of them on the superior side and the inferior side. So it's still intramural.
Yes. Actually,
not preferential
it is more
to the epicardium.
No, it's not. No preferential right now. Actually, this guy says that it can be some preferential. Well, beside the last paper, I have seen another article that actually it was a poster that I have found the paper. In that poster, the results were the earliest site were in the endocardium. So in this way, they don't say that the epicardium is the first place. Yes, according to these guys, as I understood. Yeah.
Thanks.
No, you're welcome. And yes, now I'm going to go to the automaticity. Well, the way of the automaticity is really interesting, working automaticity in the SA node. There are 2 complex clock actually that works for the SA node. One of them is the main burn clock, as they call them, and the calcium clock. The way of working is that it is somehow related to the funny currents and the T-type calcium current that starts the first early depolarization part, and after that, the main depolarization of the SA node is because of the L-type calcium currents that comes from the renin receptors and the SR. So as you can see in here, the main difference between the atrium and the SA node is that the depolarization is because of the calcium and not any sodium. And if you look at the SA node, you can see that the cardiac sodium is low in that. It means that the NAV 1.5 is low, but the neuronal sodium is really high in there. And so beside this, of course, the funny currents is the highest part in here. Well, in this way, you can say that these really change the way of the conduction to be more robust. And it can be faster than the other parts too. But SA node is highly innervated, especially in the middle part. So the changing of the different parasympathetic or sympathetic can change the place of the earliest place of the choose of the SA node. This paper is actually not all of these are regarding the human, but some of these changes are for the rabbits and the canine. But for example, for the adenosine, we can the data shows that when you place adenosine, it will goes up in the SA node, and about the acetylcholine, it will goes down. But the data of the adrenaline, as I understood, is for the rabbit. So all of these differences in the place of the conduction pathways and the place of the starting in the SA node actually is the main reason for the safe mechanism to not fail of the pacing. And in this way, we have 3 different compartment pacing and 2 to 5 conduction pathways. In this way, of course, there are less probability of fail of the pacing. And but there are some problems, of course, when you have lots of probability, the probability of the arrhythmia will goes up. Right now, the main arrhythmias that are in the regarding the SA node are the SNRT, SA node reentrant tachycardia, and inappropriate sinus tachycardia. Well, the SNRT actually can show itself in 2 ways. In the microreentries, because of the fibrosis and the fat inside the SA node, it can be inside the SA node and it will move inside the SA node. You can call it somehow a micro reentry. And the funny thing is that because of the exit block, sometimes it can show itself as a bradycardia instead of tachycardia. And this is about the micro reentries. About the macro reentries, it can happen between two conduction pathways and the difference of the refractory time of the conduction pathways. But about the inappropriate sinus tachycardia, actually the main reason, the main causal mechanism of it is still unknown. We just know that ivabradine, which will block the funny current, would control the IST, inappropriate sinus tachycardia, but ivabradine has lots of effects that are not good enough. So right now, for the arrhythmias of the IST, inappropriate sinus tachycardia, they try to ablate the SA node itself, but of course, ablating the SA node has the probability of the need to use of the pacemaker. And for the signals of the SA node, because it is an intramural signals, you cannot use the multi-electrode catheters, the normal catheters in the cath labs, but the near-infrared optical mappings shows really good. And another thing that you can understand from the near-infrared studies is that beside the different compartment refractoriness of the SA node, the conduction pathways act somehow like a filter. And it will, for example, if you pace faster than a normal speed, or even when patient is in atrial fibrillation, most of the times these conduction pathway act as an controller and filter to not to control the hyperpolarization effect of the arrhythmia or the pacing, that it will not goes to the SA node. As you can see in here, when you fast so fast, it will not goes to the SA node. Well, adenosine, because it is actually related to the L-type calcium current, can differentiate between the SA node arrhythmias and the atrial arrhythmias. And another nice thing that I understood in this way is that lots of focal crista terminalis ATs now, we can understand that these ATs are actually the arrhythmias that comes from the SA node. Because as I know, most of the arrhythmias of the AT are on the crista terminalis. And so there's another paper recently that they tried to ablate the conduction pathways, and when they done that, there is no AT anymore. So we can understand that lots of ATs right now are related to the SA node. And as I told earlier, the earliest atrial site mapping can be misinterpreted as the epi or endo. And this is not correct, according to these guys, actually. And well, I'm gonna finish my presentation right now. Actually, there are still lots of questions. I just put one of them in here. One of them could be the specific mechanism of choosing of the exit pathway. And of course, besides that, the electrode mapping is not meaningful that much, and we need another way of understanding it. And the other is the inappropriate sinus tachycardia. And thank you for your time, and I hope you enjoyed it. And if you have any question, if I can answer that, I will be doing that.
Very good, Ahmed.
Thank you,
Very
Ahmed.
good presentation.
Thank you.
Thank you very much.
Again, sorry, and thank you for your time. This was my first journal club presentation. I really need to have lots of more experience. I was talking to Dr. Cornell. Sorry that I'm taking your time right now. I was talking to Dr. Cornell that maybe choosing the review paper was not a good idea, but this is the way that now I'm working with the SA node, and it is really a nice topic, actually.
I had a question. You mentioned that the sinus node maybe has a role in ATs, and that local ablation can prevent that. Do you think that in persistent atrial fibrillation patients, the structure of the sinus node can play a role in delaying the impulse and therefore maintaining AF? Is that a possibility that you see?
According to some papers, one of the connections that goes higher it goes to the SVC or to the, some of them even, some people are saying that this goes to the Bachman bundle. So maybe these connections have some roles. I'm not sure still. This is about the conduction pathways that maybe this would affect the start of the SA node. But, start of the atrial fibrillation. But regarding the other part that I'm not sure. I think, well, because, the place of the starting of the AF in the pulmonary veins, maybe those parts are somehow related to the compartment of the SA node, that they are somehow pacemaking. I'm not sure about that. So maybe there is a connection between those, the way of these cells and myocardial cells. But
this is
I'm not sure about that.
This is why Ruben was talking about persistent atrial fibrillation, because it's important to distinguish between the trigger, which might be inside the pulmonary veins for several reasons, and what is the mechanism of perpetration. So why can't the sinoatrial node take the lead again? Is it because of a mass? It's the mass of muscle that is involved into creating the persistent circuit of atrial fibrillation, or it's because of the anatomy of the sinoatrial node? Of course, like nobody expects you to know this answer. It's just just from your informed perspective. Would it be more a matter of the fact that there is an effective exit block from the sinoatrial node, or it's because of other reasons like ion current imbalance?
Well, as I can, I think that first of all, the atrial fibrillation is somehow faster than the pacing of the SA node itself. The cycle length of the atrial fibrillation is around 180 or something like 200 milliseconds. And in this way, they are faster than the SA node itself. And as I told you, there are some studies showing that, I haven't put that picture in here, there are some studies showing that in the rabbits, when the AF is inside the atrium, conduction pathways somehow filter the AF to not go through the SA node. Of course, in this way, when the conduction pathway is not working, the SA node cannot pace again. And although the conduction part, the places of the propagation inside the SA nodes have stronger and more robust places to do the pacing, but I think they are not as strong to cancel these AFs. So this is the thing that I'm thinking on, but of course I'm not sure. Yeah.
Now the background of my question was really that in the way you presented, the SA node is a substrate, a slow conducting substrate for any arrhythmia, and independent of the pacemaker function, could act as a substrate for reentrant arrhythmias elsewhere, originating from elsewhere. So in a way, you propose a surprising vision of the sinus node being part of the AF cause rather than
well
mechanism.
Okay, actually I'm not sure about that. I don't know.
It's just
right now I cannot.
It's just how it was perceived, because it's
oh yeah, like sorry.
It participates to triggers, and the data cannot show that. No. The sinoatrial node, based on what we can see, seems like one of the many biological redundant systems. So they are built to work in any condition redundantly, so that if something goes bad, then there is another one. You have like 5 escape routes. You have very you have cardiomyocytes that do not contract. They have self-automaticity. They have a lot of supporting cells around them. They cannot be stimulated from the outside, at least from what for what we know right now. And it's premature to consider it as a potential trigger of arrhythmia. At least from the models that we have, it's not, even because we cannot even map it properly. I was wondering, since I saw that there are some EPs like Costas in the what is their opinion on mapping the sinoatrial node, or what we can do right now? Costas, if you are there, you can give us your opinion. Oh, no, it's not here anymore. Oh, that's a big deal.
Mhm.
Is there another EP here? No. Okay. Fortunately. Right. If there are any other questions, if there is any other question, please go ahead. No. Everybody's overwhelmed. Jason. Okay.
Are you planning to model this?
Well, this is what I was going to ask. Modeling the exit from the SA into the tissue, I can imagine location and conductivities and things you would modify in each branch. But retrograde conduction back into the SA node, is this something that happens? Because as we were talking about this, if it plays a role in arrhythmias, like, and it doesn't actually activate in experiments or something, how are you plan on the retrograde conduction back through the SA, through these pathways into the SA node? Is it something you'll block, or is it something I actually don't know how you do it.
Actually, I tried to model some reentries inside. As you know, right now we are working on a bilayer model, and it is a bit harder to model that. And yes, it is really hard, actually, because the only factor that now I'm working on is the conductivity. And in a way, you can say that the only factor that now we are working on is the connexin 43 and not the other parts. But there are some other parts that are working. I am right now trying to make some reentries, but because the SA node itself has lots of fibrotic tissues, and because the SA node itself has different compartment, modeling all of the parts of the conduction pathways and the SA node part is really hard. And I think making the arrhythmias in that way is really hard. And yeah, that's the way that right now I'm working on. I'm not sure if I have answered your question or not, but
well, there's an answer, because if there's a premature beat from outside the sinus node, it is possible that it can reset the sinus node. So there's no protection from the core of the sinus node from the rest of the atrium. There is a possibility of entrance block or exit block, but usually this is not the case. So usually the sinus node is being activated from an external stimulus, and then it starts again. And the phenomenon is called resetting. I was wondering, are you aware of a paper by Joyner and Van Capelle about how a small sinus node drives a large atrium? They did some very simple or basal modeling in that paper that explains how a single sinus node cell can drive the large atrium by being protected. You know it?
Well, but I would take that with a grain of salt. Since Vadim's work has come around, it's sort of changed our conception. So, I mean, you're talking about recording from the SA node. That's very difficult given the slow rate of propagation and the slow uprise. And then that sandwich between two myocardial layers on the endocardium and epicardium. So it's hard to get SA nodes. And the SA node probably starts, 40 milliseconds before the P wave on the ECG.
Yeah.
Those are quite specific structures, the exit pathways. There's probably a bit of load mismatch or something favoring direction in one
Yeah.
direction as well as a frequency filter.
Yeah.
So, yeah. So, I mean, I don't think that paper is applicable given our current state of biophysical knowledge at this point.
No, I agree. It's very simple, but it illustrates the concept of current to load mismatch. And,
Yeah, and the sino-atrial node, I mean, it's not like normal myocardium because the cells are autorhythmic, so it's better to think of them as oscillators. You have a gradient of frequency. So, depending on when the impulse retrogradely enters the SA node, it may or may not have an effect, or it may, easy say, do some phase resetting.
Exactly, yeah. So what is your objective when you're modeling this?
So what we're doing right now is we're trying to see how we can, if the P waves are altered by the exit pathway from the SA node. And we want to try and determine the atrial activation in the hopes of coming up with a better ablation strategy.
Okay. So that would
besides
the right atrial ablation strategies. That's interesting.
Yeah, so instead of, there's the factor of which exit points the propagation leaves from. There's also the question of which interatrial pathway is going to be predominant.
Yes. Very nice. Anything else? We don't.
Nothing from me as well. I'm looking forward to this. Very curious if this differential exit point can explain some P wave alternance, variability in the P wave, also in the relative activation. Curious.
Okay. There are no more questions. Ahmed, that was an excellent presentation. Thank you very much.
Thank you.
Thanks.
For June, we don't have a presenter yet. I hope we'll find one, and then.
Oh, we do have the one on the stereotaxis radioablation.
Oh, okay.
For June.
Okay, very good.
I will send you the. I think the details were shared like in January, so I will have to find them again.
Okay.
Okay.
Thanks. Well,
sure.
In that case, thank you for your attention and the presence. See you next time.
Thanks a lot.
Thank you. Thank
Bye.
you. Bye.